Hello. Again.
Hi, my name is Sierra. I'm with the Science Learning Center. I'm here to talk to you about SI. How many of you are familiar with SI? Okay, great. For those of you who aren't familiar, it's a weekly session of studying outside of class. You're meeting with an SI leader. Your SI leader is Anastasia. If you're not familiar with her or haven't gotten an email from her, you should have. You can always check out where her SI sessions are going to be. If you just Google FAU SI schedule, the schedule comes right up and it'll tell you the room, the day, the time. So these are great opportunities for you to get outside of the classroom, study some more for this class. And we do see generally better grades for students who go regularly to SI. So if you're not doing that already, we highly suggest it. And if you get a B+ or better in this course and you maintain a 3.0 GPA, you have the opportunity to then become an SI leader for this course. And if you have any questions about tutoring for other science courses or for this course, you can also email class@fau.edu and we can answer any of your tutoring questions. Are there any questions for me now? Okay. All right. Well, thank you for your time. I hope you have a great class.
Thank you.
Yeah, thank you.
All right, so I have uploaded the revised PowerPoint for today, and it's basically the same as I had last term, which was in the summer, with just a few modifications, and I did make some of the slides a bit easier to follow. Like sometimes I look back at my PowerPoints and I find there's like too much information, so I'm trying to concentrate to most important points and make it as clear as possible. Okay, so remember we have our first exam coming up on Monday, next Monday, right? That's February 2nd. All right, so we stopped last time here at the heart valves, right? By the way, I do intend to finish up with the heart between today and Wednesday, okay? Okay, so here we stopped at this slide and we're talking about the valves of the heart, right? So basically we know that we have here the atrioventricular valves, and when you look at the atrioventricular valves, you see that they are between the atria and the ventricles. So their name is just, giving away what the functions are. Okay, so you have two types of valves here. You have one valve that has three cusps, okay? And this is on the right side of your heart. So here is the right side of your heart. And you have this other valve that has two cusps, and this is at the left side of your heart, right? So your two cusps valve, that is called also your mitral valve, and is also, bicuspid valve. No, bless you. So important things that you really need to focus on the heart. So here are your valves, right? This is your tricuspid that is on the right side, and you should be able to identify which one is the right side and which one is the left side of the heart. Remember anatomical positioning, right? So here for your right side, the myocardium is not as beefy as with your left side of the heart, especially here left ventricle. Right, so important things to know here. You have the left heart, you have your left atrium, and here on the left side that you have your mitral valve, and in the right atrium you're going to have your tricuspid valve that is between your right and your left right atrium and your right ventricle. So what is the function of the valves? They basically are going to keep the direction of the blood flow going in one direction. Okay, so they are going to stop blood flow that would go back to where it came from. Right? Now, we know when the heart pumps, that flow of the blood is what gets caught by these little leaflets that are present on the valve. They work like little cup, like cells on a sailboat. So when the heart is pumping the blood and the blood has all this pressure, that puts the pressure on the cusp of those valves, and the valves will lock, blocking the flow of blood back to where it came from. Okay. Now you can imagine that pump is very powerful, so the flush of blood that is actually acting on the valve could push this valve all the way to the other side. Like the sailboat could go like break, and then the valve would break to the other side, and then you're gonna have regurgitation and a lot of problems, right? So that would not allow the blood to keep on going in one direction in the body. So in order to avoid that, the valve gets anchored to cardiac muscles that they're called papilla. So those are papillary muscles. Okay, so here you're going to have these cords, and you call that chorda tendini. So this is dense connective tissue that are like tendons. They're holding the valve to these papillary muscles on the walls of the heart. Okay, in this case here is on the ventricle, right? On the ventricles. And these are really holding the valve in place, like slowing down the closure of the valve and really avoiding the valve from opening up to the other side. Okay, so very, very important function of those chorda tendini, coordinating the function of the valves. Now, sometimes when the valve does not close properly, you are going to have a little bit of backflow of blood, back to the place, to the chamber where it came from. That is what we call heart murmur. Okay, so here I'm giving a very simplified version of the heart murmur that is only concentrating on what is happening here with the mitral valve. Okay, and this is actually one of the distinguishable sounds. The ones you hear, heart sounds, a backflow due to mitral valve malfunction or stenosis is easy to identify. So I just focus on that one as an example of an AV heart murmur. Okay, and when you look at the other valves of the heart, you see that there are many other problems that could happen, not only AV heart murmur. You could have on the left side, right? You could have on the right side, you could have aortic valve problem, right? You could have pulmonary trunk valve problems. So there are many, many problems that can happen with valves. But here I'm only giving you the example of this mitral valve heart murmur. So here is an example taken from a picture of a cadaver heart, showing you this papillary muscle that is protruding from the ventricle. Right, this is also heart muscle. So when the ventricle contracts, this little muscle here is also going to contract, and that is going to pull on this chorda tendini, and that is really going to slow the closing and prevent the flipping to the other side of this valve. All right, so here on functions of the valve, right? So it's very, very important to prevent blood backflow of the blood, and it's important to continue the one directional flow for the blood. Okay. All right, so we looked at those AV valves. Now let's look here at those outflow valves. Those are the valves that are just before the, like at the opening of the aorta, and also at the opening of the pulmonary trunk. Okay. Now these valves, both of them are tricuspid. They have 3 cusps. And the shape of those cusps, they look like half moon, so they call them semilunar valve. So you go semilunar valve. The two of them, okay? All right, so they're outflow valves. So you're going to find this one here coming from your right heart. You're going to find the valve right there before it goes to the pulmonary trunk, right, before it gets bifurcated. And here, right behind there, you're going to see your aortic valve, and it goes to the ascending aorta. Ascending means going up, right? Ascending aorta, and the valve is going to be right there at the exit of the ventricle. Okay, so that's your aorta, pulmonary trunk, right? So here is a picture showing your aortic valve. So that's looking from the top. Like, so if you look from the aorta itself and you're looking down, like towards the heart, you're going to see that the cusps here of the valve, they are facing that tube. Okay, so here's the aorta, and here are the cups. They're facing the side of the aorta here. Why? Because when the heart pumps the blood, it's going to go through here. When it gets to the aorta is a very muscular artery, so you're going to have a lot of pressure that builds up here, and that pressure that builds up in the aorta tends to push the blood towards somewhere, and if the valve doesn't close, the aorta could push the blood back to the left ventricle. So this catching of the flow of the blood here really helps to close the valve and coordinate the flow to only one path. Okay. Okay, so this is what I have just mentioned, and I really put here enough information that you can go back and review the material and study, all right? So important things to know here. Okay, here. For your outflow valves, you're going to have here the valves that go to the aorta and here to the pulmonary trunk. Now notice that inflow of blood into the atria, those do not have valves. Okay, I actually made some slides to make that clear. Okay, so let me see here. Here we go. Okay, so you don't have valves that are here between the vena cava. Okay, so you have superior vena cava, inferior vena cava, and you have openings here that go to your right atrium. So this opening that brings the blood back to the right atrium from the systemic circulation, these do not have valves. Okay, and if you look here at the veins that are bringing the blood from the lungs going to the left atrium, those also do not have valves. Okay, so we know that the blood pressure here at the vena cava is actually quite low, so there wouldn't really be how can I put this? Yeah, when you look more into the direction of blood flow, we're going to see that, you know, what is really the difference that is really carrying the blood to flow around. But basically, when the blood gets to the atria, it gets dumped in there, and once your AV valves open, the blood will drop into the ventricle. Okay, it's going to drop into the ventricles from both sides, and the dropping and the pushing of the blood is going to cause a physical closure of those openings. So even without having valves there, you don't really get backflow of blood through those vessels. Okay. There is a little typo here. there was saying about atria. Okay, right atria. No, this is right atrium. Okay. Left atrium. No. Okay, so let's start talking. Do you have any questions about the valves? No? Everybody cool about the valves? Okay. All right, so talking a bit about the veins here. So about the blood vessels. So first we're going to talk about the arteries. By definition, arteries are the blood vessels that are carrying the blood from the heart to somewhere else. So most of the blood they are carried by arteries, most of the blood is oxygenated, except for the arteries they are carrying the blood from the heart to the lungs, right? Because it's the job of the lungs to oxygenate the blood. So you actually have to get deoxygenated blood there, right? So most of arteries are carrying oxygenated blood, except for the ones that go to the lungs. And these arteries, they're actually very beefy. Okay, so they tend to be a lot thicker than veins. So they're thick-walled. They have several layers. Okay, so you have here the inner layer is made of endothelial tissue that is comparable to epithelial tissue. Okay, so it's one cell layer of this endothelial tissue, and you call that the tunica interna, so the inside mantle, inside cover. Okay, after that you're going to have a basement membrane that is going to connect your endothelial tissue to other layers, right? And after this basement membrane, you're going to have a layer of this elastic material. We call that inner elastic lamina. Okay. So after the elastic lamina, you're going to have your first layer of muscle over here. So this beefy layer of smooth muscle, right? So this is a smooth muscle that you cannot control, right? This layer is called tunica media. It's right in the middle, okay? So after this smooth muscle, you're also going to have another layer of elastic tissue, and you're going to have another layer of muscle. Okay, so all this muscle here is tunica media. Okay, now one direction of this smooth muscle is going to be perpendicular to the center of your to the lumen of your artery, and the other one is going to be going alongside. So you're going to have contraction this way, and you're going to have contraction that is kind of intercalated, okay, not all in one direction. Okay, so all this beefy part is your tunica media, and then on the outside you're going to have your tunica externa. So that is the covering of your arteries. Now, why is it that it is so important to have a very thick tunica media? Is because the arteries, they're really going to withstand a lot of pressure because they are really getting what the heart is pumping out, right? So they have to have this elasticity and contractile properties in order to first accommodate the blood that gets to them and then help to pump the blood down the arteries, right, to reach the tissues. So they have to be elastic, and they have to have their own contractile type of property. When you compare that to the veins, you see that the veins are kind of thin-walled, and the lumen of the veins, the lumen is a lot larger than the lumen of arteries. Okay, so main comparisons, main differences here is that veins are going to bring the blood back towards the heart, right? So after the arteries took the blood all the way to the tissues, so the arteries are going to get smaller, smaller, smaller. They're going to get to the capillary beds, and then you're going to get the veins. They're going to come out of the capillary beds, and they're going to start bringing the blood back to the heart. So by the time the blood gets to the veins, you don't really have a lot of blood pressure in there. So veins don't really have to deal with this change of blood pressure that we see happening with the arteries, right? So you don't really need a lot of that tunica media beefy part that you see with the arteries. So these are thin-walled, they have bigger lumen, and they mainly carry deoxygenated blood, right? Of course, except for the veins that are coming from the lungs and going to the heart, right? That's the one that carry oxygenated blood coming from the lungs, right? So they really don't have all the smooth muscle layers. They have a thin, but they do have some, right? They have a thin layer of smooth muscle, right? And one thing that is very, very important for the veins is that they have valves. Okay? They have these valves like along all the veins, especially we're going to say when we get to the veins chapter 19. So here, the reason for these valves to be present in veins is the same reason basically the valves are present in the heart and on the outgoing of the heart of the blood, okay, is to prevent backflow of blood. So the valves are positioned in a way to only allow one direction, so unidirectional flow of the blood, okay. So unidirectional, and is going to secure that the blood keeps on going until it gets back to the heart. All right. So important arteries that you should know. So this is one of the slides that really cleared up a lot of the noise that was all over this place, right? Important things you should know. Definitely, you have to know the aorta. So that's your major artery that is coming, that is going to direct the blood from your left ventricle. So here is your ascending aorta, the ascending aorta. And right after here, at the top here of the aorta, you're going to have some very important arteries that are going towards the head, right? Carotids, those are coming up here. Actually, I didn't write it here, so yeah, forget the carotids now. So here, you have the aorta, right? And you're going to have arteries here. This artery, the big one that is coming from the right ventricle and is going towards the lungs, that is your pulmonary trunk, right? And that's going to get bifurcated into left pulmonary trunk and right pulmonary trunk. So you should know that, right? Aorta, pulmonary trunk, and the coronary arteries, right? So coronary arteries, I put this extra picture here showing that they're coming from the aorta as well, and they are very important for bringing blood directly to the heart muscle. Right? So the coronary artery is going to come from the aorta and is going to bring this oxygenated blood straight into the heart muscle, because the muscle needs the oxygen and needs the nutrients to keep on pumping, right? So one thing to keep in mind here, you see you have aorta, you have pulmonary trunk, you have coronary arteries, and that goes hand in hand with the three types of circulations that you have in the blood. You're going to have aorta that is going to go to your systemic circulation. You have a pulmonary trunk that's going to go towards the pulmonary circulation. And you have your coronary arteries. They're going to go towards your coronary or heart circulation. Okay. All right. All right, so here, important veins that you should know about. Very important vein, the vena cava, right? So you're going to have the superior vena cava and the inferior vena cava. The superior vena cava is going to bring the blood that is coming from tissues that are above the diaphragm. And the inferior vena cava is going to bring the blood from everything else below the diaphragm. Okay? So they're going to come here and deposit the blood in your right atrium. Right? So besides those, you have your pulmonary veins. Right? Remember, those are bringing oxygenated blood to where? Where is the blood coming? Yes, but which part of the heart? All right, let's think about it. Oxygenated blood coming from the lungs gets dumped into the left atrium. You gotta know that, right? Left atrium. Do you have valves that are between your pulmonary veins and your left atrium? No valves. Okay. Do you have valves between those vena cava? No valves. All right. Let's see what else you have to know. Oh, and then you have the, okay, here is the one that is for your pulmonary circulation, and then this is the one that's bringing the blood from your systemic circulation, and here is the one that is bringing the blood from your coronary circulation, from your heart circulation, right? So you had here your artery, and now you have here your veins from coronary circulation that dump the blood in this big vein right there, and that is called your coronary sinus. Okay, I wish it would just be called coronary vein, so easy to remember, but no, they want to call it a coronary sinus. Right? Now the coronary sinus will bring the blood back to your right atrium. So it dumps the blood straight to the right atrium. Okay, so you see, I think it's right there. The coronary sinus is going to dump the blood right there. Does it have a valve? Is it a valve? No, no valve there, right? So this guy here dumps the blood that came from the heart tissue itself, dumps into the right atrium. Okay, first question. We got a lot of questions today. I think you have like 10 extra credit points today. All right. And, okay. All right, so question here. Why are pulmonary arteries shown as blue in the diagrams, right? So that's an easy question. Just to warm up, right? We're going to get to the difficult questions soon, okay? All right, so I'll give you 10 more seconds. It's an easy answer. There's only one answer that makes sense. Yeah, talk to your friends. Make sure you get the correct answer here. Yeah. Does anyone need more time? Is that a connection issue? All right, do quickly. Okay, I'm gonna stop it. What is your answer? C, because they have deoxygenated blood. All right, so things can go wrong, right, with the heart. A lot of things can go wrong with the heart, but one of them is problem of valve function, right? So sometimes the valves are not working well, so you call them incompetent, right? So incompetent valves is going to allow the backflow of blood, so back to where it's not supposed to go. So instead of this nice unidirectional flow, you're going to have some of that murmur thing going back. Okay. One cause of valve incompetence could be stenosis. So that is when the valves get so hardened that it doesn't open properly or it doesn't close properly, and that is due to deposits of scar tissues or tissues that are put there because of immune system functions, right? Like, have you heard of scarlet fever? Yeah, that can cause heart problems, heart valve problems, okay? So those, they can really make the valves malfunction. And if the problem gets to a certain stage, then most likely you need surgery to correct the problem. And those surgeries nowadays, they are very much routine. So you can have a valve replacement, right? And you replace it with a valve that is either artificial, so it can be just a mechanical valve, or you can have a valve that is from a different animal, like a cow or a pig, right? Those are the two most used type of valves. Or you could even have some a cadaver valve for replacement of the valve. And it used to be that before 2015 or so, these types of surgeries were very, very much invasive. Like imagine to get to the heart, you had to cut open the sternum, pull the vertebra apart, and the ribs right apart, and get to the heart and punch holes on this side and the other side. The recovery of those surgeries, the recovery was very, very painful, right? But nowadays they do these types of surgeries laparoscopically. So they punch little holes here and there, go with those little tubes, and can do everything, basically using those console game things. Like it's really cool stuff. All right, so here more on the types of circuits. That just to reiterate what I had just mentioned before, but here is in words, okay? You have a circulatory system that goes through the whole body. That's your systemic circulation. And then you have your pulmonary circulation, as I mentioned before. So here, looking at your pulmonary circulation, let me see, you have, okay. All right, so you have the blood that is going to go from your right ventricle to the pulmonary trunk, and it will reach your lungs, get oxygenated, and then they're going to come back, right? Now, when it comes back, you're going to have dumping of the blood here in your left atria. Okay. Now, okay, remember that also the blood that is flowing to the atria. Okay, let me see if that makes sense. I'm getting a bit confused now. Give me a second. Okay. All right, so the flow should actually make sense. If it happens to you that you get a little confused, like I just got right now, you go and follow the blood flow. What is it getting to your right atrium? You're getting deoxygenated blood. You're getting deoxygenated blood from your system, and you're getting also deoxygenated blood from your heart itself. All right, so that gets dumped into the right atrium. Now, oxygenated blood is going to be dumped here in your left atrium, and that is only coming from your lungs. Yeah, see, I have to redo this slide to make more sense. When you look at the volume of blood that is being pumped out of your right ventricle and is going to be pushed to your lungs, okay, that volume of blood is the same volume of blood that gets pushed out from your left ventricle into your systemic circulation, right there. This is a bit of a difficult concept to grasp, okay? Because when you think about volume of blood, first thing that comes in your head is volume of blood of pulmonary circulation is the same as volume of blood of systemic circulation. That's not true, okay? The volume of blood that is being pumped out of the heart is the same. But when you look at the volume of blood that is going around the entire pulmonary circulation, that is much less blood than the volume of the entire systemic circulation. But if you do not pump equal amounts of blood coming out from the pump itself, then you're going to have a problem of blood trafficking in the body, okay? Because these circulations, they are all in series, okay? So the flow going one way has to be the same as the flow coming back to the heart. Okay, otherwise you're going to have problems. No. So here, looking at systemic circulation, you have a lot more blood going around here than you have in the total amount of blood that goes to the pulmonary circulation. But the amount of blood being pumped is the same coming out of the heart. Okay, everybody clear with that? All right. Okay, so here is straight from your book, following one small volume of blood that gets to the heart. So here they're only showing from the vena cava for simplicity, right? So it gets to the heart, it fills the right atrium, then it gets dumped here in the right ventricle, it gets pumped in the lungs, it gets oxygenated, it goes back. So it goes back to your left atrium, goes to the left ventricle, it gets pumped out into the aorta. Okay. And yes, you do need to know the circuits, the circulations. You need to know them and the important blood vessels associated with them. Okay? Here is this very useful animation from your book. So make sure that you watch it. Who has watched this one already? One person, yay, better than none. 2, 3, all right, getting better. Okay, make sure you watch it and understand it. Okay. More questions. Which of the following is true? The left ventricle pumps more blood than the right ventricle. The right ventricle pumps more blood than the left ventricle. Both ventricles pump the same amount of blood. And I expect you to answer this very quickly and correctly. And I still am getting people giving me the wrong answer. I'm going to have a heart attack. All right, talk to your friends, those of you who are giving me the wrong answer. You got 10 seconds to correct your answer and get your points. And I just mentioned this, so if you're just like half awake, you're going to get it correct. Blood coming out of the heart from both ventricles. They should have the volume. Definitely not D. It's always somebody who gives me a D or E answer. Oh, thank you. They just changed their minds. Anybody need more time? Okay, what is your answer? C. It's the same volume coming out of those ventricles. All right, yes, somebody says it makes sense. Yes. All right, coronary vessels for your heart, right? Coronary vessels are going to feed the beef or the myocardium of your heart, okay? Remember, that is your coronary circulation, but something really interesting happens with the coronary circulation, because you imagine all these arteries are infiltrating that heart muscle. So when the heart contracts, it squeezes those arteries. That means it stops the blood flow. So when the heart is contracting, you actually don't have flow that is reaching the heart muscle itself. Okay? When the heart relaxes, that's when the blood vessels get non-obstructed. They kind of get like, okay, I was contracted when I was squeezed when the heart was contracting, heart relaxed. Ooh, okay, I relax and I let blood flow. So this cycle of squeezing the blood vessels and relaxing the blood vessels, is especially talking about the arteries, that is what you call intermittent blood flow or intermittent blood supply. So the blood is flowing through those coronary arteries only when the heart is relaxing, and you call that diastole. Okay, so when the heart contracts, you call that systole. When the heart relaxes, you call it diastole. Okay, so intermittent blood flow for your coronary vessels. All right, so here, summary locating here your arteries and veins, and here is showing your coronary sinus, and that's your coronary artery. Okay, another question, told you, got a lot of points today. What is the purpose of the coronary circulation? Okay, so let's answer quickly. Okay, 5 seconds. 2, 1, 0. Anybody need more time? Yeah, answer quickly. All right. Okay, I'm going to stop it. What is your answer? B. Yes, it's coronary, has to do with the heart. You're bringing blood to the heart. Huh? You know, I never think about the order of the answers. But that is not a good guess for these questions. You actually reminded me that one time, you know, back at NSU, the medical school, I had to submit answers to online exam, and the secretary was supposed to scramble the answers, and she forgot to, so all of my answers were A. That caused a lot of confusion. All right. So here, let's get to the challenging part of the heart, right? Electrophysiology. That's the part that scares everybody, including me. I had to study for this, okay? So yeah, I understand. I feel your pain wholeheartedly. Puns intended. Pun. Oh, I got a lot of pun for this lecture. Okay. So here, you're going to look at your cardiac muscle cell. And you know that the cardiac cells, they look like your skeletal muscle cells with some differences, right? So you see here, in comparison to your skeletal muscle cells, your cardiac cells are also striated, right? And they have those units of contraction, right? And big difference is that they cannot be controlled voluntarily. Okay, so they're involuntary muscle, and it looks like a skeletal muscle because they are striated and they are excitable. They are going to answer to an excitation signal. Okay, and both of those types of muscles, even including a smooth muscle, but they actually have to respond to a signal that we call the action potential. So has everybody seen the action potential before? I hope in some other class, some other time, yeah? So action potential is going to cause depolarization of the membrane, and then you're gonna have all the dances of the channels. They're gonna feel the change in the electrical environment, and then you have some channels that are gonna open, and then they're gonna close, and then some other channels are gonna open, and then they're gonna close, and a lot of stuff is gonna happen inside of this muscular fiber, right, this muscle tissue, and that is gonna cause contraction, right? So differences between your cardiac muscle and your skeletal muscle is that the cardiac muscle cell, which is also called a fiber, it is shorter than your skeletal muscle cell. Also, with cardiac muscle, you're going to see branching of those fibers, and you don't see that with skeletal muscle. Also, when you look at skeletal muscle, you're going to see that each of these long fibers, they have a lot of nuclei. So you have multinucleated cells. When you look at cardiac muscles, you usually have one nucleus per cell. Okay, so it's not multinucleated. Sometimes you find two nuclei, but usually you're going to see just one per cell. Okay. So cool things about cardiac muscles. You are going to have places between the cells that you call intercalated discs. So these are the dark sections, the dark areas right here between two cardiac cells. When you zoom in those intercalated discs, you're going to see a type of junction of two fibers that really keep those cells together. So that type of junction, you call it a desmosome. Okay? So this is like a button that is sewn together from both sides with these fibers that are going to keep those cells side by side. And that is very important because, you know, heart is pumping all the time and with a lot of force. And if the heart pumps and the cells are not very much attached, then those junctions could rupture and you could have a broken heart, right? Like literally, right? So you don't want that to happen. So those desmosomes are very, very important to keep the heart cells stuck together, okay? And you're also going to find between those cardiac cells, you're going to find this little straw, is a little tunnel, okay, from one cell to another. You call that gap junction, okay? So they have an opening that connects the cytoplasm of these two cells. So both cytoplasms here, they can talk to each other. And this is very, very important because when one cell undergoes depolarization, it is going to send this electrical signal right through these gap junctions to the next cell. So both cells are going to feel the depolarization together. And then all of the cells are actually connected through these gap junctions. So a whole section of your heart, either on the top or on the bottom, a whole section of the heart is going to do the response to the electrical signal. It's going to do the response together. Okay, so these gap junctions are really what are going to be responsible for spreading out the action potential from a group of cells that received it to all these other cells that are connected. Okay. All right. So here is another picture showing the desmosome. Okay. So here is plasma membrane of two cardiac cells. So here's a desmosome, and here you have your gap junction. Okay, gap junction that is going to connect the electrical signals. And here you're going to have your thick and thin filaments that are going to respond to that action potential and shorten and cause contraction. Right? And all this thing here is happening in those intercalated discs. Okay. All right, so this part is really, really cool because here is showing only like a fraction of the heart, is really showing the area where you have those AV valves. Okay, so you have here your, where's the beefy side? I think is on this side here. You have, oh, these two valves are kind of looking like not the way you're supposed to, but it's only a fraction. What is the idea here? Between the atria and the ventricles, you have your AV valves, right? And here you actually have around those AV valves going all across the heart, you have what they call the fibrous skeleton of the heart valves. And this is mainly made up of collagen, and collagen does not allow for electrical signals to go through. So this part of the heart is really an insulator of electrical signals. So when you have an action potential that happens here at the atria, it is going to stay only at the atria because this insulator here is going to block the transmission of that electrical signal to the ventricles. All right. So all of these cells from right and left atria, all of those cells are connected by those gap junctions, right? So they're all going to respond to one electrical signal and they're all going to contract together. But this electrical signal is not going to jump through the directly through the contractile fibers to the ventricles. It's not going to happen. This blockage here is going to maintain the contraction separated from the ventricles contracting. Okay, so it's really working as this electrical firewall, right? Then later on you get to how the impulse is going to get from the top to the bottom. That's the later. Okay, so having this type of cytoskeleton here, this type of collagen blockage really separates the heart into this area that is all connected, and this area is all gonna contract together, and you call that a functional syncytium. Right there. Okay, so functional syncytium for the top part of the heart, both of your atria are going to contract together, right? Then you're going to have some fibers here. They're specialized in taking the signal from the atria and bringing the signal down to the ventricles. And here they're going to excite cells that are going to contract in the ventricles. So once the cells start to contract here with the action potential, then all of these cells here in both ventricles, they are electrically coupled and they are going to contract at the same time. Okay, so here is going to contract and then it's going to relax. So what you're going to have, you're going to have the upper part of the heart contracting all together, then relaxing. By the time these guys are relaxing, these guys here all start to contract together and then relax. So now you have your pump. It's like boom, 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 boom. So all of the cells are going together. Syncytium here, syncytium there. Do they ever take a break? No. Otherwise, you die. No break for the heart. Another question. Told you, lots of questions today. All right. The presence of intercalated disc. Presence of intercalated discs. The questions are getting more difficult. Are you going to see these types of questions on the exam? Yes. Right. And gap junctions between adjacent cells cause the heart to behave as what? Okay, 5 more seconds. There you go, E. Okay, change your mind. All right, does anyone need more time? Quickly, quickly. Okay, put in the answer. Okay, everybody put in the answer, yeah? Yes, all multiple choice. Test is all multiple choice. Okay. All right, what is your answer? D. They work together. Functional syncytium. Okay, so similarities of heart muscle with your skeletal muscles, right? Continuing here. you do have in both types of muscles, you have this resting membrane potential that is at a negative value, right? Let's say about negative 90 millivolts or so, okay? So here, when you're talking about the heart fibers that are capable of contracting, okay, the heart has two types of fibers. One of them is contracting fiber, okay? The other one is going to be called pacemaker cells. But these guys that we're looking at now, those are the fibers that contract, okay? So these guys, they're going to receive a signal and they're going to depolarize. When they depolarize, first channel that gets open is a voltage-gated sodium channel, just like with the skeletal muscle, right? So voltage-gated sodium channel opens, floods the cell with this positive ion, right? Then you're going to have here calcium channels that are going to open and put calcium inside of the cell, right? And then you're going to have calcium that is going to bind to troponin. Remember the guys? Where is it at? Is like connection, is like blocking the attachment of thin fiber with thick filament, something like that. Thin filament, thick filament. Okay. Calcium binds to troponin, and then you have muscle contraction, and your fibers will your muscle cell will shorten. Okay. Big important differences here, okay? So this is very, very important for the heart, okay? After your initial sodium influx, right, so that's your depolarization signal, you have these calcium channels that open, so this is T-tubule, right, same thing that happens with the muscle. Your calcium channels are going to open. These guys are very fast, okay? So after they open, you're going to have also opening of another type of calcium channel that is right here, okay? So this is also at the plasma membrane, okay? And these guys are slow. You call those calcium channels L-type. I would call them slow type. So the L is for slow, okay? So they are going to open, and they are going to allow even more calcium to get to the cytoplasm, okay? Besides that, besides those two, okay, because this, by the time this slow guy opens, it's also the time that that potassium channels start to open. Okay, so let's see what happens here with the calcium first. So you're going to have this wave of calcium that gets in here is going to increase the concentration in the cytoplasm up to a point that allows binding of calcium to this other calcium channel that is present in the sarcoplasmic reticulum. So this calcium channel right here, when it binds to calcium, it's going to open and is going to allow calcium to come out of the sarcoplasmic reticulum and go also to the cytoplasm. Okay, so this type of action here is what we call calcium-induced calcium release. Okay, so calcium-induced calcium release of your of the calcium storage that is in the sarcoplasmic reticulum. Okay, so this channel here, you call it ryanodine receptor. Okay, this is the ryanodine receptor. So three types of calcium channels. You have your fast calcium channels that feel the change in the voltage and they open right away. You have your slow calcium channel that feel the change in the voltage and they take their time to open. And then you have your ryanodine receptor that is in the sarcoplasmic reticulum, and those are going to depend on calcium getting there for the channel to open. Okay? All right. So what is happening here with the action potential in the heart muscle? That's the part that is really different when you compare with the action potential in the skeletal muscle, right? Because the skeletal muscle didn't have all these different types of calcium channels. It just had one type of calcium channel, okay? So what is happening here? First thing that happens, when you get here your action potential, first thing that's going to happen, your sodium channels open. influx of sodium. So your membrane voltage goes from negative all the way to positive something. Okay, so inflow of sodium. Then you're going to have here opening of calcium, fast calcium channel. You're also going to have opening of your potassium channels. But you're going to have, as soon as your potassium channels are opening, you're also going to have opening of your slow L-type calcium channel, right? And then you're going to have opening of your ryanodine receptor channel. So you're going to have a lot of calcium that gets dumped into the cytoplasm. And the positive charges that are brought by calcium to the cytoplasm, they counteract the action of the potassium that is trying to leave the cell. Okay, so when you look at your membrane here, let's put a membrane here. Okay, so you have sodium going in, you have calcium going in, so you have like a lot of calcium here, and you have even calcium from SR, and then you have potassium trying to go out. So we have like one charge potassium trying to go out. When potassium is able to go out, that's what's going to do the repolarization of your membrane. But your potassium is trying to go out, but a lot of these other positive charges are in, so you are counteracting the action of that potassium going out. So you end up keeping the potential of your membrane up in a positive side for a very long period of time in comparison to your muscle, skeletal muscle cell. Okay, so this is what we call the plateau of the action potential of your heart contractile cells. Okay, and that is due to the open calcium channels. Okay, so the open calcium channels, let's compare it with the sodium channel. Okay, so sodium channel at first, it opens when it gets to a certain threshold voltage. And then when the membrane becomes positive, what happens to your sodium channels? What happens to the sodium channels? They stay open like forever? No, they are closing by inactivation. Remember the little ball on the channel on the cytoplasmic side? Yeah. So sodium channels get inactivated. Now, something similar is going to happen to the slow calcium channel here. So this low calcium channel will open and then is going to get inactivated. Okay, so here is a calcium channel, this low guy right there. Okay, this little guy right here. It gets inactivated by calcium binding to an inside part of the channel. So the channel will also get inactivated. Yes, you have a question? No? Stretch? Okay, it's good, stretch. Okay, so calcium channel, slow guy, is also going to get inactivated. When the calcium channel gets inactivated and does not allow any more flow of calcium, that's when your potassium is going to take over and you're going to start to repolarize your cell. Okay? So here you have sodium action, calcium action, which is very long plateau, and then repolarization by potassium leaving the cytoplasm. Okay? Now, this whole extension here under this plateau area is really going to make sure that all of the cells that are supposed to contract together in syncytium, is going to give it a chance for all of the cells to contract together and make sure the contraction is complete. And then they're all going to relax together, okay? And really make sure that you don't get extra contractions in the middle where you're not supposed to get, okay? So what this thing is doing is really making sure that your absolute refractory period, that means the time when you cannot contract the cell again, right? Your absolute refractory period is quite long for your cardiac cells, okay? So that ensures that you're not going to have one part of the syncytium contracting here, another part contracting there, and they're not coordinated. That would be a mess, okay? So making sure that doesn't happen, you have here your very long refractory, absolute refractory period. Okay, so here you see this graph showing the force of contraction. So after your action potential, you start contracting everybody gets contracted together, a lot of force, and then everybody starts to relax right after repolarization. Everybody cool with that? All right. So here, continuing with the differences, okay? Remember, your heart cells have the inactivation of your potassium channels, and those are the L type, okay? These are the L type. And your refractory period is quite long, okay? And besides making sure that you have the syncytium and maximum force, this action of everybody contracting together also avoids what we call tetanus. So tetanus, when you look at skeletal muscle, you see a totally different way of contracting, because the skeletal muscle is going to respond to your will of how much force you want to use, right? So let's say that if I want to, push this microphone away, I go, well, I'm activating these many muscle fibers. I don't need a lot of force. So I send a signal through a nerve that goes to the neuromuscular junction and tells my arm to push this a little bit. So I'm not activating all of the muscles that I could possibly activate to do the action, right? But then that means you have an option that instead of pushing this, if I am in a very bad situation, like, let's say a good thing, like if I'm taking boxing lessons, right? I'm punching a beanbag. And I feel like on a day that I'm kind of frustrated, I have to study all the stuff that I don't understand half of it, so I need to let the frustration out. So what do? Punch the pillow, punch a beanbag, let the energy out. So let's say now I want to punch this imaginary beanbag, and I go like, okay, if I go like this, the bag is now going to move, so I got to go there and punch it. Now you're going to recruit a lot of muscle fibers together, right? That also means that if you want to exert a certain force and you are recruiting a lot of muscle fibers, you can contract a lot of them together, like when you are at the gym doing weightlifting, right? That's one kind of thing I don't like to do. But let's say that you're really trying to get those weights, like the guy, the thing that goes like behind your neck, and you go like this, getting those muscles, and it's a lot of weight, so you're pushing, you're trying, you're not moving much, and like, oh my God, this hurts. You are putting a lot of strength in there, and you're contracting all these fibers, and continuous contraction, that's what you call tetanus, right? But it's because you have certain fibers contracting and other fibers relaxing, and they are exchanging their functions so that you can keep that muscle contracted for a long period of time. That cannot happen in your heart. Otherwise, you die. Simple. Anything goes really wrong with the heart, and you die, right? So this mechanism of very prolonged total refractory period and coordinated syncytium contraction, those are really safety mechanisms that avoid tetanus and avoid fibrillation. I don't even know which one is worse. Oh, they both kill you, so I guess they're both bad. Okay? So coordinated contraction, long refractory period, and this is really, the long refractory period is really due to your inactivation of those slow calcium channels. Okay? Gotta avoid tetanus. Okay. All right, so here chart straight from your book, comparing the two types of muscles. Okay, so here is your beautiful action potential, so showing these stages, right? So here is your sodium influx, and here is your calcium and potassium with extended calcium plateau, And this repolarization is really due to your potassium channels, because potassium is going to flow from the inside of the cell to the outside of the cell. Potassium efflux right there. Okay, and here just for the last slide, another point, no, other points for you. All right, the plateau portion of the heart action potential is due to what? And yeah, that's the type of question that you're going to find. Right. All right, so remember what you got to know about this action potential, right? What kind of channels are opening, what kind of channels are closing, which are the channels that inactivate, and then for how long, and then what causes the closing of the channel but not inactivation of the channel, right? All right, does anybody need more time? No, everybody answered? Okay, what is your answer? B. B. Okay, here we go. Okay, so yeah, I thought wouldn't have enough time, but hey, I finished 9 minutes before 6:50. Yay! Okay. You have